Today we will be talking about lipids. This is the last video over lipids. I know it was a large chapter, but lipids are a huge biomolecule within our bodies, so it's pretty important for us to understand how lipids operate. I will first talk about cholesterol and its effects on the cellular membrane. Okay, so essentially cholesterol, we'll call that chol. Well, it's actually a bulky, rigid molecule. And rigid. So why is it rigid? Well, it's rigid because it has a lot of cyclohexanes in its structure. Okay, so imagine at least 12 cyclohexanes paired together, and then you have some other R groups on the chains. Well, that's going to offer some stability and some strength, right? And because it's so rigid and so hard to break down, it actually controls how fluid the membrane is. Okay, so we can say controls, and you can say instead of rigid, you can say how firm or how strong the membrane is. Controls how firm or strong the membrane is. And of course, chol means cholesterol. Right. So how does that happen? Well, it's kind of counterintuitive, but here's how I try to explain it. Remember how we have our polar heads and then we have our fatty acid tails? Let's imagine that we're talking about triglycerides. Okay, so just a storage unit for fat. Well, normally, if you pack them together like this, you have a very firm structure, right? They can't really move around too much, and so the cellular membrane becomes very firm and it can't move around. And if you have too many saturated fats, the cellular membrane cannot move and transport as efficiently. So you can't get in nutrients as efficiently. This can lead to heart disease or other bad diseases in your body. And so what cholesterol does is that this is without. So we're going to put without cholesterol, and then we're going to put with cholesterol. You're going to have this. So we still have our triglycerides, which are unsaturated, but then let's draw this. This is going to be a cholesterol, okay? That's a triangle. The triangle is cholesterol. So what cholesterol does is says, okay, well, what if I wedge myself between these triglycerides and try to separate them as furthest as I can? Well, that's pretty cool, because now you can actually have some fluidity. These saturated fats can't be as packed together, and therefore they cannot make the cellular membrane as rigid. Okay, so this is actually a good cholesterol. Okay, so this is actually a HDL. So that's the one that is good for you. You shouldn't really be avoiding cholesterol in your diet, because without the little cholesterol molecules in the cellular membrane, your cells would be too rigid. They wouldn't be able to transport nutrients as efficiently. This one actually increases the fluidity of the membrane. So we can say that cholesterol, cholesterol right here, let's do it here, chol increases fluidity of the, and yeah, we'll just call that cell mem. Okay, so cell membrane is going to be MEM. Okay, so again, the cholesterol wedges itself between the saturated triglycerides, and it makes the cell more fluid, which is good. So as you can see from this little table that I have, we can see that the cellular membrane is made up of two molecules, mainly two molecules. It's made up of lipids and proteins. Now remember in the first video that I made, I said, hey, the cellular membrane is comprised of lipids and fats. Well, here you have the mathematical version of what I said. Sometimes you have all three. You have lipids, proteins, and carbohydrates within a cellular membrane. But protein, so we call that proteins, are about 20 to about 80% of the cell membrane. Okay, so there we go. And the rest is usually made of lipids and carbs. But the thing about proteins is that they're distributed unevenly across the cell. So let's make up a cell first. So proteins are distributed unevenly on the cell. So let's make up a fake cell right here, and this is going to be our proteins. So our proteins, let's say that we have a certain protein right here, and there's one right here, there's one right here, one right here, and then there's only one over there. So notice that a majority of these little triangle proteins are going to be on the left hand side, and only one of them is going to be on the right hand side. Well, why is that? Well, sometimes, let's say that there's a membrane over here, which is in teal, and the cell is between the membrane. Well, what if this has water on this side and this has no water? Well, you can tell that these proteins are going to be hydrophilic and they want to be on the side that has water. Well, this one is also hydrophilic, but it's not really going to want to be on the side with water, but it's still there because you have to have some proteins on both sides. So what I mean by uneven distribution is that sometimes within membranes you have proteins that want to be on the outside because they're hydrophilic, and then you have some proteins that are hydrophobic and they're going to be on the other side. But on both sides you're going to have either type of proteins. It's just how it is, okay? Just how it is. It's weird. But they're distributed unevenly across the cell, okay? And before we get into it, we actually have two types of proteins. We have two types of proteins. And essentially, the two types are integral membrane protein. Well, pro is for protein. And then we also have the peripheral membrane proteins. Now, we're going to be talking about the integral membrane proteins in the next slide. So, now we have integral membrane proteins. What are they? Imagine that this right here is the lipid bilayer. Okay, so you have a cell and the membrane is going to have a protein that goes on the outside environment into the inside environment. Okay, so this is going to be inside the cell, and this is going to be outside cell. Okay, now we're focusing on this protein right here. Well, why does that matter? Well, integral membrane proteins, which we're going to be called IMPs, we're going to call them IMPs, this abbreviation, right? So the reason why this is so important is because they actually allow molecules in and out of the cell. All right, pretty interesting. And the reason why, well, not the reason, but because they allow molecules to be transported from within the cell to the environment, or from the environment to the cell, they are called transmembrane proteins. They're sometimes called transmembrane proteins. Okay, what do I mean? Well, let's say that you have glucose. Okay, glucose is a simple sugar, and if you've taken physiology, you know that glucose needs to enter the cell in order for the body to work properly. Well, let's do a little glucose. Let's make that a square. Well, actually it's going to be on the outside. So what happens here is that glucose isn't really that polar. It's not that polar. It doesn't know how to get into the cell by itself. But what happens now is that the glucose is going to use this as a highway, kind of like a little tunnel. And it's going to go in here, and then it's going to come out into the cell. Okay? Well, is it going to do that if the membrane of the protein is polar? No, it's not going to happen. So what I'm trying to say is that integral membrane proteins actually have a polar side and a non-polar side. And we can actually see this using a 3D model. So this is called the beta barrel motif. Okay, so this is the beta, let's do that. This is the beta barrel motif. And it's actually the same thing. It's just a membrane protein, but it's made of beta barrels, or excuse me, beta sheets. So it's important that you know that this is going to be this is anti-parallel beta sheets. Okay, and the hydrophobic sheets. So you remember how beta sheets can be made of hydrophobic or hydrophilic amino acids? Let's say that the beta sheets inside the protein are hydrophobic. So internal, so we're going to say internal beta sheets are hydrophobic. Okay, so you have your amino acids that are hydrophobic. You have the leucines, alanines, isoleucines, right? And they're going to be inside. So the glucose, whenever it passes through here, whenever glucose passes through the membrane, is going to go here and is going to be making contact with the hydrophobic amino acids or beta sheets, okay? And let's say that this is going to be across the membrane. So here's your cell membrane, blah, 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 right? So we have cell membrane. Well, on the outside surface, on the exterior, you're actually going to have your hydrophilic amino acids, okay? So exterior has hydrophilic. That's an H, amino acids. Okay, so there you go. And where do you find the beta barrel motif? Where do you find this? Or where do you find integral membrane proteins? Well, they're usually within bacteria. So found usually in bacteria. Where else can you find this? You can also find it in the outer membrane of the mitochondria and chloroplasts. The outer membrane of mitochondria and chloroplasts. Because why? Well, mitochondria, they need molecules in order to do reactions, right? They need molecules to make ATP. Chloroplasts need molecules to make energy for the plant, okay? And so these little beta barrel motifs, these integral membrane proteins, are used within the membrane in order to do transportation. They transport molecules from the outside of the cell into the cell, and from the inside of the cell to the outside environment, okay? So you can think of these as little tunnels. Pretty cool. What else can you do with integral membrane proteins other than transport molecules? Well, they actually act as receptors. So we can also say acts as receptors. So within your brain, they can send messages in and out of, different cells. So if the cell signals that there is too much glucose in your body, well, it's going to release a message from the integral membrane protein. Okay, so let's say that the square in this case is a message, and it's going to get out from the cell, and it's going to follow a pathway to the brain, and it's going to say, hey, there's too much glucose, we need to do X, Y, and Z in order for the glucose levels to go down. Well, in this case, the integral membrane protein acted as a receptor. It actually allowed the message to go on, okay? And you can see that within homeostasis and set points, okay? So if you've taken physiology, you know that if your body goes away from a set point, let's say that the set point for the body is about 97 degrees Celsius. If it gets too hot, so it's 99 degrees Celsius in your body is going to release messages to your brain, and it's going to signal a reaction. So you're going to start sweating, and that maintains homeostasis. But that is only possible with the use of integral membrane proteins. What is a very famous integral membrane protein? Well, one of the most famous ones is the alpha helical membrane protein. Okay, so this is called the alpha helical. So it's just like an alpha helix, okay, membrane protein. All right, what is it? Well, imagine an alpha helix going between the outside and the inside of the cell. All right, so that must mean that it could go once, let's do this in green, can cross the membrane once or many times. Imagine that you have just a strand, okay? A strand of spiraling amino acids. So like alanine, leucine, valine, they're just all repeating and repeating, and they go in and out and in and out of the membrane. Well, how long does that have to be? Well, they're usually about 20 to 30 amino acids long. And if you were to measure it with a meter stick, let's say, let's just joke, like a meter stick, then you would actually measure a length of about 30 angstroms. So that's like 1 times 10 to the negative 12 meters, okay? So they're very, very small, and they go in and out of the protein or of the cell membrane at least X amount of times, okay? They're about 20 to 30 amino acids long, okay? So why should we care? I mean, it doesn't seem too important to me, but that's where you're wrong. Why? In humans, okay, the alpha, let's say MP, okay, let's just say alpha MP is helical membrane. In humans, alpha MP is in about 27% of proteins. So in your body right now, 20% of your proteins are actually made from alpha helical membrane proteins, okay? So, like, it's major. You have to know about this. You have to know that they facilitate the transport of what? Of molecules, of nutrients in your body. 27% of your proteins are made from that, right? So on the exam, if they tell you, what is, or if they ask you, what is a major integral membrane protein in your body, you're going to say alpha helical membrane proteins. We will now move on to the peripheral membrane protein. Not a lot that you need to know about this guy, but you still got to know about him. What exactly is it? So it's a little baby protein that interacts weakly with the lipid polar heads. So we're going to call them PMP, peripheral membrane protein, interacts with the polar heads of the lipids. So remember, this is like the phosphate group, and these are going to be the fatty acids, okay? So we can say the lipid polar head. But it's very weak, so it's a weak interaction. Weak interaction. What kind of interaction is it? Is it covalent interaction? Is it ionic? Well, it is actually hydrogen bond, so H bonding interaction. And also electrostatic interactions. So why should we care? It doesn't look that important. It just looks like a blob to me. Well, blobs are pretty important, okay? Because this blob right here can act as an enzyme. It can act as a signal transduction protein, so it can do signals within membranes, okay? And it can actually become a cytoskeleton proteins, so they can provide structure to the overall cell. Without these little boys, you wouldn't be able to function. Your cells would just be like a blob. They wouldn't have a shape. With these guys, they form a shape and they keep that shape. They can also make signals between cells, okay? And of course, they can lower reaction rates because they're enzymes. So can be enzymes, signal makers, and cytoskeleton proteins. And I guess you can think of it as superglue, right? So you're trying to build something. Are you going to use like a bunch, like a whole gallon of superglue on a project? No, that's too much. Superglue is really powerful. You just put a drop on something and it sticks forever. Well, it's the same thing for peripheral membrane proteins. You just put a drop here, a drop there, and everything just comes together quite nicely, okay? But in the same way that it's like superglue, you can also get rid of the glue, right? So you can put like a detergent in the superglue and it's gonna unbind. Similarly, you can put a detergent on the membrane and get rid of those membrane proteins, okay? I remember when I told you about snake venom. Well, the phospholipase A2 is going to break down some of the fatty acids in your body, and then it's going to turn the fatty acid into a detergent, and then the detergent is going to attack the blood and it's going to make it pop because the blood doesn't maintain its shape. Well, when the detergent attacks the cell, it's actually attacking the peripheral membrane protein, okay? So whenever it attacks this guy, the blood pops, okay? So, I mean, they're very important. Without this guy, your cells wouldn't really exist, okay? So that's why we should actually know how they operate and why they're so important. You can also get rid of these guys with salt, just normal salt. And let's say you're doing an experiment, you can put salt into the environment and it's going to whisk away the peripheral membrane proteins. And then you can also decrease the pH. Actually, sorry, increase the pH. So you can actually say to remove, we can increase pH or salt concentrations. And we can only do this because the interactions that it makes with the lipid polar heads are very weak, okay? Now let's bring everything that we learned in the past 2 videos into fruition, okay? So we're gonna bring everything together and it's gonna make a nice little picture. This is actually the fluid mosaic model. This is the fluid mosaic model. Imagine that everything is moving at once. So you just have a whole swarm of squiggly things, okay? And they have the consistency of vegetable oil. So this isn't necessarily firm. Imagine that it's like a liquid inside and it's always moving around. Remember how in the last video I told you that these polar heads are always kind of moving side to side. Sometimes this polar head, right, or I guess this lipid might go this way and end up here in like 2 minutes or something. Sometimes it might hit a flipase and go over here to the other side, but that takes a lot of energy and it's also a slow process. So normally they move side to side. Okay, well, sometimes we don't want the membrane to be so firm, right? So we're going to introduce cholesterol. And right here, you have these little yellow things that go between the lipids and prevent them from packing so tightly. So these are going to be unsaturated. Let's assume that this is an unsaturated saturated cell membrane, and because of the cholesterol blocking the distance, they're going to be not as packed tightly, and you can have some movement. If the cholesterol was not there, you wouldn't have any movement at all, and that would be pretty bad for the cell membrane. Okay? Now we're going to introduce the integral proteins. So this one, let's say we have a glucose molecule coming from the outside, and it wants to go inside. So let's put it over here. And he's going to go here into the non-polar region and come out here into the polar area. Okay? So it acts like a tunnel, like a transporter. What kind of integral protein is this one? Well, on the exam, if they show you a diagram, you should be able to tell me that this is the alpha helix. Okay, so this is the alpha helical membrane protein, and you can tell because it has these little baby hairs on it that I like to see, or call them. Okay, and sometimes this might go back into the cell again, and it might come back out and go in, etc. It might do that a lot of times. You don't know. What else can we say? Well, there is a peripheral protein right here. Okay, and that acts like an enzyme, and it acts as a supporter for the membrane. So you can pretend that it's like a little hook that it attaches to like the cytoplasm, right? So I think that is pretty cool. Okay, and of course, how fluid the membrane is dependent on temperature. So if it's too hot, the membrane is going to be more fluid, and if it's too cold, the membrane is going to be more rigid, more saturated. Okay. Another thing that can dictate how fluid the membrane is how long the tails are. So how long the fatty acids are will dictate how fluid the membrane is. So short fatty acids make it more fluid, but longer fatty acids make it more rigid, more structured. Why should we care about cell membranes anyways? Well, we should care about cell membranes because they prevent the cytoplasm. So let's say right here we have cytoplasm from leaking out of the cell. So we prevent the cytoplasm from leaking out. Okay, again, you want your cells to have a shape. You don't want them to just be like a splat inside your body. If you don't have these cell membranes, you don't have a container for the cytoplasm, which has a lot of proteins in it and a lot of fats that's good for your cells. You don't want that leaking out into the environment. Okay. Another thing is that cell membranes, they actually allow for the transport of molecules. Okay, so without the cell membrane, you wouldn't be able to put potassium in your cells. You wouldn't be able to put sodium in your cells. So they allow for selective selection. Well, it's weird, but selective permeability for molecules. Okay, so these are the reasons why cell membranes are so important. Again, cell membranes can actually act as receptors, so they can send messages to other parts of the body, and that's important for us. Without that, we would have been dead a long time ago. All right? And if you've taken physiology, you would know how ATP is created, and essentially you have a membrane over here, and in this membrane you have these, I'm not really going to go into it, but you have these complexes, and you have your electron transport chains, right? So we have NADH. This isn't important for you to know. So it just touches this on the cell membrane and it makes hydrogens. And then the hydrogens are going to go over here to ATP synthase. And you're going to have ATP. You have a bunch of other steps and different electron transporters, but that's the main gist of it. But what I'm trying to say is that without cell membranes, you would not have that. If cell membranes did not exist, you wouldn't be able to generate ATP. You wouldn't have electron transport chains. And so this all ties in to animal physiology, human anatomy. It ties into pigs. Without cell membranes, you wouldn't have baby pigs. And so I think that's one of the main reasons why we should know about cell membranes. So whenever you're questioning why you're taking this course, think of the baby pigs. Let's pretend that you're babysitting and you're making about $12 an hour per kid. That's a pretty good job. But now the kids are getting hungry and you have to feed them spaghetti. Well, when a baby is eating spaghetti, how does the nutrients from the spaghetti get into the cell? pretty simple. First of all, you have a glucose molecule. Now let's pretend that it's a whole piece of spaghetti, that it didn't digest properly. Okay? And it's going to enter the cell membrane via the integral membrane protein. Okay? So it's going to go into the non-polar region and come into the cell. Now whenever a molecule goes into the cell in one direction, okay, you have a, you actually have a uniport. So uniport is only for one molecule. Okay? That's the only thing that matters. It doesn't matter the direction. It could go into a cell, it could be coming out of a cell, it just has to be one molecule. However, the direction does matter when you're talking about symport and antiport. So symport means that it's for more than one molecule. Okay, usually it's about 2 molecules, very common. And that means that it's 2 molecules that go in the same direction. In the same direction. You may have some books that say symport is only when the molecule goes into the cell, and antiport is when the molecules come out of the cell, but I don't like that. Okay, I don't like that definition. I prefer the definition of direction. So if they go in the same direction, whether it's inside the cell or out of the cell, it's going to be symport. But antiport is whenever the molecules go in the opposite direction. Okay, so that makes more sense to me. One of the most prominent antiports example would have to be the sodium-potassium pump. Okay, it's a very important pump that regulates your cells. Without it, you wouldn't be able to generate ATP or glucose. Okay, and an example for symport would actually have to be the glucose sodium diffusion. Okay, so yeah, so now you know about symport and antiports. There's about 3 steps for a molecule to enter and exit a cell. So let's say that this glucose molecule touches the entrance. Okay, so let's say that these 2 cylinders are closed. Whenever the glucose touches it, it triggers a reaction, and the molecule or the membrane is going to change shape. Okay, and then the glucose is going to go into the cell and do its thing. But as it's leaving the membrane protein, it triggers another reaction that closes behind it. Okay, so it's like a door. So the glucose touches it, it triggers a reaction, it opens, the glucose comes into the cell, exit the membrane, and then as it's exiting, it triggers another reaction that forces this membrane to close. Okay, so there are 3 steps. So we call that binding, aka touching, change shape, so we could call that open, and then release, which is close. Okay. Now let's talk about some diffusion. Okay, so for most people it may be a review, okay, but I'm required by the United States government and education board to cover this material. Okay, so here we go. This is going to be passive diffusion. And let's call that diff for diffusion. Now there are 2 types of passive diffusions. Okay, there's simple diffusion, and there is facilitated. Okay, this one requires no ATP. It means no energy needed. Why is that? Well, you're relying on the concentration of the outside compared to the inside. Okay, so right here you're going to have a higher concentration of, let's say, sodium ions. Okay, and then you're going to have your membrane, which is like this little wafer thingy, and there's too much energy here, right? Because sodium ions, they move around a lot, and whenever they move around, they generate energy. Now, the system doesn't like having too much energy on one portion. It likes to split up the energy evenly. So therefore, the sodium ions from the higher concentration are going to naturally diffuse across the membrane to make an equilibrium. So let's say that there are 50 sodium ions here and 50 sodium ions here. Over here you had like 90 sodium ions and about 10 sodium ions. It doesn't like that. So essentially higher concentration, higher con to lower and it happens spontaneously. So when I mean spontaneously, I don't mean fast. This could take hours to do. By spontaneous, I mean that it happens naturally. It doesn't need any energy to do it. It doesn't need any persuasion for the reaction to be completed. It just does it naturally, okay? So that's going from a high concentration to a low concentration. But what about facilitated diffusion? Well, take everything that I just said and put it into an example of a red blood cell. So over here we have 5 millimolar of glucose, okay? That's a lot. Inside the red blood cell, there is about less than 5 millimolar of glucose. So let's say that we have $5 here and we have about 10 cents inside the red blood cell. Well, we have a lot more glucose outside than we have inside. So we want the glucose to go inside the red blood cell. However, the membrane for the red blood cell is not very cooperative, right? So glucose can't just get into the cell easily. It needs something else. It needs a permease. It needs a protein tunnel. Okay, so you can think of it as an internal membrane protein. So it uses internal membrane proteins as a tunnel in order to get into the system. And so essentially what facilitated diffusion is that it uses internal membrane proteins or channels in order to allow diffusion. So you're still relying on the fact that there's more stuff outside than there is inside, but you're incorporating a tunnel, essentially. It doesn't need any ATP, it does it naturally, and those are the two types of passive diffusion techniques. And finally, we will be talking about the active diffusion. Okay, so this is active diffusion. What this means is that you have the opposite of passive diffusion. So you have sodium that goes from a lower concentration out of the cell into a higher concentration into the cell. Okay, so let's say that there are 50 sodium ions inside the cell, and there's only about 2 sodium ions outside. Well, you want the sodium ions from the outside to come inside, you see. So it goes from low, so we put goes from low concentration to high concentration. Okay, so that is not easy to do. It actually takes up energy, requires energy. And this energy actually comes from the form of ATP, okay, adenosine triphosphate. Cool. There's also another type of active diffusion. So we call that, well, this is actually an example of primary. So this is going to be primary active diffusion. But there's also secondary active diffusion. What is secondary active diffusion? Imagine that these sodium ions go into the membrane. So 3 sodium ions go into the membrane, and as it's entering, it makes an opening. Now glucose sees this opening, and he says, well, you know, might as well enter at the same time that the sodium enters. And therefore, it enters in a Symport fashion. What I'm trying to say is a secondary active diffusion uses ATP just so that the first molecule could get into the cell, and then the second molecule, in this case glucose, piggybacks off the door opening. So an example of this would be, let's say you forgot your key card to enter a building. So one of your friends uses their key card to open the building or open the door, and they let you in as well. You didn't have the energy needed to get into the building. You needed another person to use some energy so that you can both get in. So secondary active diffusion, so we have secondary active diffusion is when a second molecule uses the first molecule's energy to enter the cell. Okay? And again, this is a very prominent example, the sodium potassium ion pump. So 3 sodium in, and then you have 2 potassium out. Okay? And of course, there is an enzyme that allows us to do this. It is called the sodium potassium ATPase. ATPase. So it's an enzyme. Okay? And that's all we're covering in lipids today. So lipids are done for this chapter. And I hope that this video helped you understand everything you need to know about lipids and that you do well on your second exam. Remember that I believe in you and that you're putting all the effort into studying. And thank you so much for spending your time watching these videos and for just believing in yourself. So have a great day and take care. Love you.